Hi, everyone. Thanks so much for coming. Oh, cool, it works. So I'm a fish evolutionary morphologist, which means that I study the shape or the geometry of anatomical structures in fishes. And I'm going to focus mostly on my lab's shark research today, but I also wanted to shout out my amazing grad students. All but one work on bony fishes, but it's okay. Including Nicole Rahman, who just defended her thesis last Friday, so congrats to Nicole. Thanks. Everyone's working on different aspects of anatomy or evolution of largely local species right here in Humboldt, including larval fish, fish that have been separated by the Klamath Dam complex, olfactory morphology of trout, which is what Nicole worked on, or even glue production by some of our local species while they're mating. And you'll see Natalie's work a little bit later in the talk, so no spoilers. Okay. But the core of my lab's research is not bony fishes. It's this group, the cartilaginous fishes, or the chondrichthyans. And this is a little tableau by Ray Troll that shows you the diversity of body forms in this group over the last 400 million years of their evolutionary history. This is a group that's really captured my imagination because of this morphological diversity. And they do all of this with a really interesting array of body plans, ecologies, and morphologies. And again, morphology really is just shape of their anatomical structures. But when I talk about, oh, our lab studies sharks, you probably think of like Jaws. And yet, they're ecologically really diverse as well. So you have the largest species are filter feeders like the basking shark. There are scavengers like sleeper sharks. This is the cookie cutter sharks. Anyone heard of that thing? Okay, so they're small, but they take like flesh cookies out of dolphins and whales, or submarines, which was a big problem during the Cold War. Or they could be an ambush predator. This is a California angel shark there eating a horn shark, so he's pretending to be sand. And then he's going to engulf that little horn shark, which gets its name because it has spines on its back, so then it regrets its decision. But sharks also don't just have an array of diets, they also occupy a really wide range of habitats. And they have what I consider to be superpowers. So all sharks have pores on the undersides of their noses that allow them to perceive electrical fields produced by other animals. There are deep water sharks like Edmopterus that actually have little photophores on the undersides of their bodies, and even ones that can walk on land. So this up here is the epaulette shark, moving from tide pool to tide pool. But one of their most amazing, superpowers is actually related to the detection of low-frequency sound. So sharks perceive the low-frequency sounds that are produced by struggling prey, but also those produced by waves crashing on shore. And so to understand this, let's think a little bit just about how hearing works. Obviously, this is a human ear, and we have an outer and a middle ear. So our outer ear stops at the eardrum. We have middle ear bones, and then this is an inner ear that's shared by all animals with backbones. And sound waves vibrate through this system and ultimately end up vibrating fluid that's in those little membranous canals. We can render out shark inner ears from CT scans. So just like getting a CAT scan at the hospital, we isolate those ear shapes from the insides of shark heads using digital technology. So this is our local salmon shark, Lamna ditropus, and I'll render the cranium transparent so you can see in pink there those ear canals of their inner ear. And if I blow that up, this is just a bunch of anatomy here, but they have a couple of really recognizable specializations for picking up low-frequency sound. Like this back semicircular canal there is isolated from the other two structurally. Oops. And the whole ear is actually walled off from their brain cavity. So those are two things that are easily recognizable for us in these CT scans that show us that they're picking up low-frequency sounds. And when we study the anatomy of fossil sharks, so what you're looking at here is a 326-million-year-old brain cavity and inner ear from a species I got to name after my graduate advisor, and he was really embarrassed. Back then, they actually did not have these specializations yet. So this is a shark, and yet this inner ear is just confluent with the brain cavity. So we can look for these adaptations early on in the shark's geologic time and figure out when they started picking up low-frequency sounds. But even recently, folks have just started to look at the diversity of ear shape in living cartilaginous fishes, living chondrichthyans. There was a study just 2 years ago that assessed about 2% of living cartilaginous fish species, and they found that the size of structures of the ear correlated with things like diet, and habitat. And yet, ears didn't originally evolve for hearing sound. They show up first in invertebrates for an organ of balance, for a sense of equilibrium. So in this model here, this person's semicircular canal is detecting how they move their head. These detect roll and pitch and angular acceleration as that fluid sloshes around in your ear canals, okay? So you can thank that for your motion sickness. So if your ears are perceiving how your body is positioned in space, then it's natural then to assume that the shape of those ear canals might correlate with habitat, with how you move through your environment. And indeed, this has been found to be the case in every other vertebrate group besides fishes so far. So this is an example from salamanders, where the shape of their inner ear canals correlate with life history strategies and also with habitat, for example, with those that burrow. So this makes sense if you're inhabiting a three-dimensional environment like burrowing underground or living in a tree, you're going to be picking up that angular acceleration or rolling different from an animal that just walks along the ground, right? So we think that might be the case in sharks. If a shark is living in a reef habitat that's really three-dimensionally complex, maybe it's going to have a different ear shape from a shark that lives along a sandy bottom or in blue open water. So we're starting to do just that. We recently got NSF funding to survey all of the morphological diversity of shark inner ears. This is the basic workflow. We look at CT scans and isolate the inner ear, which is like coloring for adults. It's really fun. We generate what are called landmarks that capture variation in that shape, and then we plot them in what's called morphospace, and we correct for the degree of relatedness. And our aim is to answer the question whether shark ear shape is driven more by phylogeny, which is how related are they to each other, versus ecology. So do you have a particular ear shape because your ancestors had that ear shape and so did their ancestors? Or do you have a particular ear shape because it's useful for the habitat that you live in? That make sense? Okay, cool. So thanks to a bunch of amazing undergraduate students, two of whom are here today. So shout out to Casey and Isamar, who have done really the bulk of this work. We have already surveyed almost every living family of living cartilaginous fishes and generated those 3D models of the inner ear. And then Natalie Swearingen, who's over there, is going to explore these connections between anatomy of the ear and this, what we call phylogeny, a diagram of evolutionary relationships. So that question of, do they look like that because they're related, or do they look like that because of their ecology? And here's just some of our early data. So the ears are in pink. We're looking top down at skulls of sharks with the noses pointed up towards the top of the screen. You can see they vary a lot in size and shape already. And if I plot that on one of those evolutionary tree diagrams, and I also want to mention this is an undergraduate-led paper, so Casey's first author on this paper, Isamar's on this paper. They're going to present their work at meetings, and hopefully this sucker's going to get published. It's in review. So when we look at these evolutionary relationships, yeah, there are some similarities in all these little ear shapes, especially in some groups, that maybe show us it's phylogeny. Maybe it's relationships in evolutionary time that are driving this. If I overlay some habitat data on here, then it's like, well, maybe the pelagic stuff looks more similar, but it's hard to explore that qualitatively, right? That's where we need those quantitative analyses. And if I overlay diet data, there's not really much rhyme or reason to it. And so this has, for me at least, lent some support to this idea that maybe it's not listening for struggling prey that's driving ear shape in this group. Maybe it's much more about the habitat that they live in or their phylogenetic history. And I think this is exciting. If we do find that ecology and ear shape are correlated, that might let us predict some more about the habitats of extinct shark species. Then if we have a well-preserved ear, like the one I showed you earlier, are we able to look at that now and say, well, maybe this was a shark that was inhabiting a reef versus the pelagic habitat, or maybe just sort of humbly on the sandy bottom. Thanks very much for your time.